So Josie has headed to Aslo, so she asked me to kind of moderate before I do Anne's intro. So does anyone have announcements before we start? No, it's Easy Friday. Okay, so today's talk is from Anne McElroy, who is professor and our illustrious graduate program director here at SoMAS. For the past 30 years, her research has focused on factors influencing bioaccumulation, metabolism, and the effects of organic contaminants in aquatic organisms. Before coming to SoMAS, Anne came from Chicagoland, as she puts it, and then went on to get a BS in aquatic biology from Brown. She then did her PhD at the MIT Hui joint program in Woods Hole. And then did a postdoc at the EPA laboratory in Narragansett. Before she came full-time to SoMAS, she served as a director of the Sea Grant program and as a faculty member in environmental sciences at UMass Boston. So I started working with Anne as a postdoc in 2010, which she says is the highlight of her career. And on some of this work that you'll see today on Winter Flounder. So with that, here's Anne.
Thank you. All right. That's good. Can everybody hear me back there? Okay. Well, I'll try to do this without spilling coffee all over my computer. Maybe I'll move my computer because I took out my last computer and spilling beer all over. I was celebrating this grant, actually. I was happy that day. Anyways, I wasn't sure what to talk about. It's been a long time since I've given the seminar here. It's so long that I actually can't remember how long it's been. So that's like way too long. So I decided to talk about some of the work a bunch of people who I've been working with have done on winter flounder in urban estuaries. And really, hopefully, it's a snapshot of what fish in urban estuaries have to deal with. So what do we mostly do in my lab? I'm sure most of you think I just harass you about graduate program stuff. But in my spare time, I try to do other things. And the work in my lab is mostly split in between kind of two approaches. One, going out into the environment, and Long Island is a wonderful environment to do this because we've got lots of gradients in population density and contaminant exposure, all kinds of things. So part of my work focuses on collecting animals that are naturally exposed to different environmental conditions and different types of chemicals and bring them in the lab and study them. So-called natural experiments. And then, because it's often hard to get to these places, particularly in winter, and it costs a lot of money for ships, I have kind of a parallel set of things going entirely in the lab where we use mostly naive fish, mostly these days fish embryos, and maybe in a couple of years here at the Shubham talk about that in his dissertation, to evaluate more mechanistically the effects of chemicals on organisms. But today I'm going to talk about the field-based work, and specifically studies done on young of the year winter flounder. I'm going to give you an overview of three projects. The first, a study on endocrine active compounds in Jamaica Bay. This was a work conducted by a graduate student of mine, Lourdes Mena. The second is a study on immune competency in winter flounder in Long Island bays. This was a master's thesis by Jamie Romany, who worked jointly under me and Mark Fast. And then hopefully, if I don't spend too much time on that, I'm going to talk about our current project, which is the most interesting and most ambitious, as you'll see, a multi-parameter assessment of winter flounder condition in Long Island bays. And this is being run jointly by Lyndie, who we hired to run the whole thing, Mike Frisk and Mark Fast and I, and along the way we've constricted a coast of cast of thousands, it seems, people working on different aspects of this, and I'll just show you snippets of some of their work. So winter flounder flounder is the ideal organism. They're sweet, they're docile, they're easy to work with. They're not so easy to find when found, but it's a great test species for evaluating the effects of sediment-sorbed contaminants or local conditions. They live on and in the sediment, so they're intimately exposed. They eat benthic organisms that eat and live on and in the sediment, mostly worms and amphipods. So they have kind of maximal exposure. They have limited migration patterns. So they have this on-offshore winter-summer migration, spending half or more, it turns out, some of them don't migrate so much, it seems, of the year in shallow embayments where they also spawn. So they spawn inshore where contaminants are more likely to be in higher concentrations. The eggs are benthic as well. So their young are exposed from the very beginning to the full suite of things in sediments. And best yet, all the young fish actually stay where they're born for at least the first year. So you have a pretty good idea of what kind of conditions they've been exposed to. Unlike many fish species, particularly pelagic ones, that go here and there and you don't really know where they've been. Winter flounder is also important because it's commercially and recreationally fished, and stocks in the Northeast, and particularly Long Island, have been in decline. And data indicates that young of the year survival may be limiting recruitment. And just to demonstrate this, these are some data from the southern New England, mid-Atlantic Bight, winter flounder population, commercial landings, and I can do this two hands. You know, you see there's some peaks, but since the mid-'80s, you know, pretty steady downward trend, and things don't look so good right now. They look even worse in Long Island bays, and this is some data Mike put together from the New York DEC seining survey, going from 85 almost to the present, and you know, the last 5 years particularly have not been good years for winter flounder. Now, to set the stage for why we started out looking at inactive substances in Jamaica Bay is some earlier work done in my lab and done collaboratively with Bruce Brownwell, looking at sewage-derived contaminants. And STPs here, sewage treatment plants in New York, we determined put out effluent that can feminize fish using juvenile hybrid striped bass. We found that younger fish were more sensitive than older fish, not surprising. In studies with juveniles bass, it took 3 weeks to get measurable effects. In newly hatched fry, we could get effects in 96 hours. Turned out the most estrogenic effluent, the effluent we've studied 4 different plants, was distinguished by having very high levels of this group called nonylphenol ethoxylates, NPEOs. And these are detergent breakdown products that turns out are pretty good estrogen mimics. And Bruce also found that these NPEOs are found at really high levels in Jamaica Bay and elsewhere, turns out Hempstead Bay, at levels as high as tens of parts per million in the sediment. And he also determined that in anoxic sediments, as many of these organic rich sediments are, they get preserved. So our hypothesis was very simple. Fish living in urban estuaries may be feminized, possibly by these NPEOs. And this work was kind of inspired by a growing controversy developing at that time that there were a lot of compounds out in our natural waters that maybe shouldn't be there. And this Science News article here, I love this swimming in steroids, you know, after this cruise ship debacle last week. Maybe we could have even worse ones. But this came out in 2001, and this was a news follow-up on a large reconnaissance study that the U.S. Geological Survey did, measuring a whole suite of polar compounds, hormones, other pharmaceuticals in surface waters from some like 200 sites around the U.S. And what they found was at many of the sites they could detect, albeit very low, levels of hormones and drugs of all different types. Now, just because they're there doesn't mean they're causing damage. This article over here was from Newsday. That came out, I think, in 2007. And that had to do with the fact that it wasn't just hormones we were worried about. Many common use household items like detergents and things carry substituents that can also be hormonally active in the environment. So there was increased concern about endocrine active chemicals in inland and coastal waters worldwide in the 90s and 2000s. So that really sort of inspired this work. So the objectives of the first study I'm going to talk about were basically to assess whether or not winter flounder in Jamaica Bay, a place where Bruce had determined had such high levels of these compounds, are presenting evidence of endocrine disruption, and specifically to determine the influence of these nonylphenol oxalates. And we did experiments on non-spawning adult fish. We did some trial experiments on embryos and larvae. But it turned out we did most of our work on the young of the year juveniles, in part because we really couldn't find enough adult fish to do any significant studies on. And today I'm only going to talk about the work on the juvenile fish. So here's our study sites, Jamaica Bay over here in the west, and the blow up of that, and we used Shinnecock Bay as our reference site. And as all of you who've studied Jamaica Bay know, it's we often fondly call it a sewage estuary because most of the fresh water coming in is sewage effluent. There are 4 plants depicted here with S. The blue stars there are study sites. And this area over here, right by Kennedy Airport, I want to point out, this is Grassy Bay. It was dredged when they made the airport. It is a high deposition site of really fine sediments. These are where the highest concentrations of these nonylphenol oxalates have been measured. And it was sort of the focus of our study. Before I go on to talk about the effects, I just wanted to give you a little bit of background about why we were measuring some of these things. By the way, this is really kind of an overview talk. I am not really going to present any methods at all. There isn't time. If anybody wants to talk about the details of them, I'd be happy to answer questions or talk to you about it later. Hopefully I'll be able to make myself clear without doing that. Anyways, vitellogenin expression is one of the most commonly used measures of feminization in aquatic species. It's an egg yolk precursor protein, and it's induced in response to estrogen, usually in spawning female organisms. Although all fish can express this from a very young age, and males can express it too, they generally don't express it at very high levels. So if you see elevation in vitellogenin levels in juvenile organisms that are sexually mature, or males, it's seen as an indicator of some kind of exposure to estrogenic compounds, either estrogen itself or an estrogen mimic, and it's usually measured in plasma as the free protein. Another condition that's often followed is something called ovotestes. This is the presence of oocytes in what would normally be male gonadal tissue, and it's often observed in response to estrogenic compounds. And then finally, sex ratios. Female biased sex ratios have also been observed in response to estrogenic chemicals, not surprisingly. One thing I'd be remiss in not pointing out here, fish kind of are free-form spirits in the universe, and all these things vary quite a bit, species to species, and what's normal may not be what you would think is normal. For instance, there have been studies showing high levels of ovotestes in some fish in various areas, even really biased sex ratios, and actually some fish can change their sex as needed. So all of these measures need to be, and an absolute number doesn't really tell you anything. They are all measured in reference to some unexposed population or some controlled population. So, my saying a 50-50 sex ratio, that it happens to appear to be the norm for these winter flounder, but it may not be the norm for winter flounder elsewhere or some other fish. So, what did we see? This is the first data year we produced in 2002, and it's showing vitellogenin in fish plasma, micrograms per mil. as a function of site. And in this first study, we measured vitellogenin by ELISAs. And the 4 sites on the left are all in Jamaica Bay. This is Grassy Bay, Floyd Bennett, I forget what EP stands for, they're on the other figure, and this other one. And this actually is an average for several sites in Shinnecock Bay, our reference site. But the point is, we don't see big differences, 2.5 to 4, but we do see statistically significant elevation of vitellogenin in these fish as compared with Shinnecock Bay. Now these fish in the first year were captured in May and June, so they're fairly small and fairly young. And in this slide and almost all the others, I'm going to present means plus the 95% confidence intervals. Okay. We went back in 2003 and 2004, this time getting older, larger fish. And the same thing happened, only a much bigger response. Note here, this is now a log scale. So again, Shinnecock Bay, in these years, we only focused on Grassy Bay. Big induction of vitellogenin in both years in the Grassy Bay fish as compared to the Shinnecock Bay fish. We also, we're not histologists, but we did a little bit of histology, and these are some micrographs showing a testes with this ovotestes condition. It should look, this is a very good slide, but it should look speckled here, normal testicular tissue. It should not have these little donuts in it. We also saw evidence of testicular degeneration in these fish. And most interestingly, we saw highly skewed sex ratios. So the fish from Jamaica Bay, albeit not a huge sample size, we saw 1 male for every 9 females, as compared to Shinnecock Bay, where every time we looked, we had a dead-on 1-to-1 relationship between males and females. I didn't show you this data, but part of the reason we had to abandon the earlier plan to use the adult fish is we really couldn't find any male adult fish in Jamaica Bay. They just weren't there. Of course, there weren't a lot of adult winter flounder in Jamaica Bay, so it wasn't conclusive. So moving on to the second part of that first study, we wanted to see if nonylphenol was likely involved in any way in this. So first off, we did a preliminary study where we took some reference sediments from Central Long Island Sound, and we dosed them with nonylphenol, the most potent member, at a concentration of 50 micrograms per gram, the highest concentration we actually saw in Jamaica Bay. And we had these cute little winter flounder condos up at Flax Pond, and we had 8 winter flounder. This is a study of 8, preliminary study, where 4 of them individually got exposed to sediments dosed with nonylphenol, and 4 of them got just exposed to the reference sediment. And lo and behold, 15 days later, we see almost identical levels of induction of vitellogen to what we had seen in the field. But as I said, this was 8 fish. I wasn't going to stake my career on this. So fortunately, hooking up with Chris Chambers at the Sandy Hook Nymphs Lab, where they have beautiful aquatic facilities, we got him some adult fish from another study I was doing with Henry, and he bred them in the lab, and the leftovers from his experiment he graciously decided to let us do some experiments with. So we scaled up this nonylphenol experiment, exposing the fish in duplicate tanks in these nice troughs at Sandy Hook to either sediment from Grassy Bay, diluted sediment from Grassy Bay, because we were worried it would be completely toxic to these fish. So we did a 1-to-4 dilution with reference sediment, or reference sediment dosed with this nonylphenol at the same concentration we used in the last study. And we also had some fish either in reference sediment alone or water alone, because we weren't sure how well they really handle sitting in buckets of sediment in the lab. We had 20 fish per treatment. We did all the treatments in duplicate. We exposed for 4 weeks. And we measured plasma vitellogenin and sex ratio. This is looking a lot like that last slide, where here too, in a much bigger experiment with much better numbers, we found significant elevation of vitellogenin, 2 levels observed in the field caught fish in the non-phenol dose sediment, and in both the Jamaica-based sediments, the grassy-based sediments, the diluted one and the non-diluted one, as compared to the reference sediment. And again, this is a log scale. That part worked really well. The sex ratio part did not work so well. Unlike previous years where a month of growing would have gotten these fish to a size that was large enough that we could histologically sex them, most of the fish that we harvested at the end of that 4-week experiment were not large enough to sex. So we only got a very limited number of fish that we could accurately determine whether they were male or female, and those data are reported here. So there's a hint of a reduction in the percent of males, so a feminization of the grassy bay fish and the non-phenol fish as compared to the reference fish with, by chance or not, still reporting the exact same 50-50 sex ratio that we saw in the field collected fish. But unfortunately, as I said, these numbers were too small to show any kind of significant statistics there, which was really disappointing because that was a huge effort. So the take-home messages from this Jamaica Bay endocrine study are presented here. We saw elevated vitellogenin, altered sex ratios, and presence of ovotestes in Jamaica Bay as compared to Shinnecock Bay fish, indicating endocrine disruption with potential for ecological significance. The experiments with nonylphenol showed pretty conclusively, at least I think, that the observed levels of nonylphenol in the field are sufficient to cause the feminization we saw, but they don't prove that they were causing the feminization we saw because there are myriad other chemicals there as well. Okay, the second study, this was Jamie Romany's study done jointly with Mark Fast, sought to evaluate the interplay between contaminant exposure and disease susceptibility, again in young adult flounder. And this diagram, although it's not very good, I should have borrowed a better one from Bassam. It's one you've seen many times in the studies coming out of his lab. This is the famous disease triad, which tries to depict how properties of pathogens, the disease organism, properties of the susceptible host, what that disease organism is going to attack, as well as properties of the environment that can influence both these factors, form the conditions ripe for the occurrence of disease. And so what we were trying to do in this study is look at immune response factors in winter flounder, again from the field, and also try to look at biomarkers of contaminant exposure in these fish to try to see if more urbanized fish had any differences in their immune susceptibility. So the hypothesis I just stated not so well is right here. And what we set out to do, or what Jamie set out to do, is measure antimicrobial activity, so an actual functional activity in mucus scrapings off the surface of these fish, and also look at expression of genes involved in the antimicrobial peptide pleurocidin as another measure of immune response. And at the same time, she set out to measure cytochrome P451A as a biomarker of contaminant exposure. And for those of you, many of you probably who are not interested or aware of cytochrome P451A, this is the predominant enzyme involved in metabolism of a whole suite of contaminants, as well as steroids, different forms of it. And it's been used as a biomarker of exposure to things like oil, organic contaminants, PCBs worldwide for decades. So this is what people often measure, and we thought we would measure it too. So these are Jamie's study sites. We wanted to include Jamaica Bay again and Shinnecock, but then she, piggybacking with the DEC survey, had fish from Oyster Bay, Manhasset Bay, and Little Neck Bay, hooking up with Mike's people. We got some fish from Port Jefferson Harbor. So she had these 6 sites and was hoping to look for differences in these immune parameters. So here is her data on antimicrobial activity. This is a Vitrometric assay, you look for light associated with microbial growth. And she looked at 2 different species of Vibrio, Vibrio angularum and Vibrio parahemolyticus. And presented here is the percent inhibition of that bacterial growth normalized to milligram of mucus protein. Right. Not really striking patterns here. Oh, these are the different sites from east to west here. Shinnecock, Port Jefferson, Oyster Bay, Manhasset, Little Neck Bay, and Jamaica Bay. Lots of variability in the data, evidenced by these box plots. The letters show which of these groups are statistically significant from each other, and there are very few. I mean, you could, if you're leaning right, imagine an east to west gradient here. Certainly Shinnecock is different from Jamaica Bay here. It's also different from Oyster Bay, but you know, I wouldn't want to bet the bank on that. And that's only for one of the bacteria. The other one, there was absolutely no trend with site. Here are the data for pleurocidin expression. And this was in thin tissue as well as cytochrome P450 expression, and this is in liver tissue. Again, nothing here, and this was not what we predicted. Something's going on at Port Jeff here, but there's certainly no east to west or west to east trend here in either of these parameters, which was a real surprise. Now, to fill you in a little bit on the mystery of why Port Jeff has such extreme variation, it turned out that pleurocidin expression, at least in these young developing fish, seems to be a function of both size and age. So if we separated the fish into smaller ones, of which were most of the fish we had, and larger ones, so this is 76 and this is 7 for those who can't read it, making the cutoff at 125 millimeters in total length is the break off between what we call small and large fish. And you can see there's a significant difference with the bigger, older fish expressing more pleurocidin. So maybe this immune competency is just something that comes with age, and these really young fish just can't do it. Here's all the data, and you can see how it slips out. Guess where all these big fish come from? They're feeding at Danforth. These are all Port Jefferson fish. They were probably right off the dock there, or maybe off those big yachts that like carpet. Anyways, so you know, it's an interesting thing. It would be interesting to follow up on this. Jamie graduated and left, so she's clearly not going to do it. But it points out the importance of taking age and size into effect, because for some of these parameters, we see age-related effects. For others, like CYP1A, we don't. So the take-home messages from her study was there did not seem to be the hypothesized large urban gradients in altered immune status, nor CYP1A expression from the bays she looked at. But the individual variation in all the parameters we looked at was so large, and in some cases was influenced by size and age. You can't really separate them in these young, fast-growing fish. You know, maybe really these were not the most sensitive things to be looking at. And all the work I've talked about so far is limited in scope in a couple important ways. These experiments were designed to test fairly narrow hypotheses, measuring a limited number of endpoints, focusing on biochemical and cellular reactions in individual fish. And although from things like that we can document exposure and effects, and we can infer potential ecological significance, we don't, that's all we can do. So really what's needed is more comprehensive studies examining a more robust set of endpoints, enforcing factors, alongside with more ecologically relevant endpoints. And when Mike Frisk joined our faculty, it was like, yes, we're going to team up and do this together. So the third project, which we are just rounding to a close right now, set out to do just that. The goals of the project were to investigate factors influencing the survival of young of the year flounder in Long Island bays, looking at both biological parameters, more ecological biological parameters, things like predation and population structure, and also health or condition, as well as environmental factors such as temperature, oxygen stress, and this perceived urban gradient. And for this third study, we concentrated on the South Shore. We included Jamaica Bay in the west. We added in Hempstead Bays because of the group project going on there. We added in Mauritius Bay because it's at the foot of the Forge River, and there's been a lot of good work recently done there. Shinnecock Bay, Cold Spring Pond, just because it was convenient and it was there, and Napeague Harbor for a variety of reasons that we won't go into right now. And here are the overall project components. The environmental measures, oxygen, temperature, and salinity, population surveys to determine abundance and size that allows us to estimate mortality and growth rates. We did a cage study to do a predator-free, a predator exclusion study, so we could look at survival in the absence of predators and growth that way. And then we did a whole suite of measures on individuals. We did condition index, we did hepatic somatic index, we looked at muscle RNA DNA ratios as a proxy for recent growth. We did muscle total lipid as a measure of energy reserves. Brian dissected out otoliths and to analyze settlement date and daily growth increments. Shannon took fin clips we did and looked at population genetics by microsatellite analysis. And we dissected out the livers, or should I say Lyndie, and Shubham a little bit, dissected out livers to do gene expression analysis. And the whole goal here was to take all that data at the end from a 2-year study and with Mike's help, put it into a comprehensive model using multivariate statistics to try to look for patterns and association between all these things. So I'll start with population surveys. We did beam trawl surveys every other week from May through October. I actually did some of these, Lyndie didn't do them all. 10 random stations per day at each site at 1 to 2 meter depth. We did 5 sites in 2010. We went back to 3 of those in 2011, and we also sampled in Hempstead in the second year using seines because it turned out we just couldn't get any fish with the beam troughs. Here's the kind of data we could get from some surveys. So catch per unit effort was calculated for each sampling date, and then these catch curves created. This is the first year and the second, and the regression is used to calculate the mortality rates. And those are shown here. This slide that Mike put together compares our data, which is the blue bars and the yellow bars to the right, with data previously reported by others in Connecticut and Rhode Island. And in this slide are some earlier data from his lab, 2007 and 8, that Melissa Yensho did her master's thesis on, as well as the data from our 2-year study for all the sites. The error bars are only included on the Shinnecock Bay sites because it would make this slide just really messy. But what you can see from here is there's a lot of variability. There's some year-to-year variability. But overall, we don't see clear differences in mortality between the sites we were investigating, although we do, overall in Long Island, seem to have slightly higher mortality rates than other people have reported. This describes the cage study. At U, Jeffrey, Lyndie, and a team of thousands, it seemed, built 12 1 meter by 1 meter by half meter cages, 4 for each of 3 sites. We went out and caught every winter flounder we could get that day, and were able to deploy 6 to 9 young of the year per cage. And we deployed them at a 1 to 3 meter depth, depending on what the tide was doing. Along with the winter flounder on the bottom, we had these sondes attached to one of each of the 4 cages at each site. And while they were out there, we were able to continuously record temperature, salinity, and dissolved oxygen every 30 minutes. And this is a picture shooting up from the bottom of the cage. Are these 3/8 inch mesh? What's the mesh? quarter-inch mesh here, and you can see how small these flounders are. There's one of them. But I wish I had a picture of this, but the way we measured growth in these things is Lyndie would pull them out and then get underneath the dripping cage with a camera, shoot up, and then knowing the size of these meshes, we could figure out the size of the fish, which was actually really masterful because getting fish in and out of these cages was well, in the limited time they're out of the water, would have been no easy feat. So that was great. That's why she always wears sunglasses in the view. Anyway, so I'm just going to show you some highlights of this stuff. Hypoxia seemed to be the most closely linked to mortality in both the field and cage fish. And here's data from Jamaica Bay. Here's our entire SOMAS record of dissolved oxygen. And the red line here is at 3 or 2? 2. 2. And in this box here, this is where it kind of bottomed out, and where our mortality data from the individuals in the wild caught fish. So these are different sampling dates, and the fish in the cages always seemed to drop when we started to get sustained hypoxic conditions. And in this case, at least in Jamaica, the fish in the cages experienced somewhat a delayed in mortality, which might suggest some refugia from predators at this site. But if you look at Mauritius, which are shown in this figure, again, the box here corresponds to where we had the live fish data. You can see here too, this is where the populations are crashing both inside and outside the cages. Here there didn't seem to be any cage refugia going on. And then in Shinnecock, much better dissolved oxygen conditions overall, but even there too, at the very end of the summer, it started to dip into hypoxia. Yeah?
What is your coordinate over there, the left-hand axis?
It's, Lyndie's going to have to explain that one. It's
On the bottom
the fish numbers. It's numbers, log numbers per.
of the catch per unit effort. So it's the way that we do it to calculate the mortality curve.
So is that between 2 times, or what? I don't understand.
Each time we
It's a
went
measure of
out there,
So it's the catch per unit effort is basically number of fish per pulls of the net or something like that.
right, so we did 10 trawls at each site every time we went out, and that's where these come from. So overall, Shinnecock had much better conditions with respect to dissolved oxygen, but did have some excursions into low DO. And actually, this is the only time we saw a dead fish in one of these cages, indicating that it had died just prior to us getting there, because once these things die, they're just eaten up and gone almost immediately. Okay, so we also measured a lot of indices in the individual fish, and these are summarized here, where, I'm sorry you can't read this very well, but it goes from Jamaica, right? I didn't read it, yes. So going from Jamaica in the west to Napeague, the site out at Montauk. And the way these plots from our work, the little notch part here, demonstrates the edges of the 95% confidence interval. So if the notches don't overlap, you can infer statistical difference. But you don't need to infer that here, because basically pulling all the data across sites for any of these indices, you don't really see much in differences. I mean, Hempstead Bay here, for this RNA DNA ratio is a slight reduction there, but not much going on here. Of more interest are the temporal trends in these condition indices, and for virtually all of them, we see dropping off in the late summer when conditions become really hot. For the condition index, Fulton's K, the hepatosomatic index, the lipid content of the muscle, and interestingly, for the RNA DNA, we had the most excursion here of values. We see the drop, and then we start to see it recovering somewhat at the end of the season. These are data that Brian Gallagher here worked up on otoliths after dissecting, what, 300?
260.
260, almost 300. Really small fish to get the even smaller ear bones. I couldn't do this. I couldn't even see them with my eyes. He did micro-otolith analysis, which allowed to determine settlement date and to measure growth rates of all the fish. And here is the data for 2010, where we had 5 sites, and 2011, where we had 3 of the same sites and 1 additional site. And don't have time to go into detail on this, but what his data showed is prolonged spawning and differences between spawning times of the sites, but fairly similar growth rates overall, and those data aren't on the slide, between both year and between the sites. But strong seasonal cycles in growth rate were evident. These are work and beautiful slides made by Shannon. Shannon's probably at the Shark Conference, showing the microsatellite data analysis, which we did to look at population structure in these communities. And this is the FST value that shows relatedness of population. And as you can see from the statistical table at the bottom, I mean, you can't see because it's too light. This is Shinnecock over here, and it was different, statistically different from every site other than Cold Spring. And Napeague also was different from the sites other than Jamaica for some reason. So what this is telling us is there is micro population structure even among these populations in the South Shore. And that is particularly interesting when the paradigm has all their parents mingling offshore and then coming into spawn. Now, even more interesting is this determination she made of internal relatedness, and you're going to have to talk to her to explain exactly how this is done, but we all like this slide so much because it put things in perspective. A completely outbred population would have an interrelatedness factor of 0, and if you had clones or twins, the value would be at 1, so it ranges between here and here. The overall mean for all our fish was 0.226, which puts them at the rate of half siblings. And if you look at the individual populations here, you can see where they fall out, with Shinnecock being kind of average, Hempstead and Jamaica actually being on the low end of interrelatedness, and Mauritius and the Napeague fish being at the top end. So, but the rest of the time left, I'm going to just briefly talk about the gene expression work, because it was probably because it was the part that I was most directly involved in. But based on the previous studies, particularly Jamie's work with immune response and gene expression, we didn't want to spend 2 years going after specific target genes that we hypothesized would change, only to find, after a lot of money and a huge amount of effort, that we didn't really have patterns in anything. So rather than do that, we decided we'd look to the flounder itself and let us tell us which genes are likely to be changing between sites. And so with the recent affordability of high throughput sequencing, we did embark on a little sequencing project with these winter flounder. So what we were looking for is whether there was any kind of transcriptional signal associated with flounder condition and demographic patterns. Potentially looking to identify biomarkers associated with their bay origin, and also to provide a data resource to others evaluating winter flounder gene expression. So we used this relative expression method where we collected, there we go, livers from late season young of the year fish from 2 sites which we hypothesized had different condition but were closely enough related that should have been the only differences, Mauritius Bay and Shinnecock. We extracted RNA from individuals, We pooled those individuals because even though it's much cheaper than it used to be, these are very, very expensive analyses. And so we had a pool from Shinnecock, we had a pool from Mauritius, and then using an Illumina platform, we did pair and repools, our RNAseq, to completely sequence these 2 pools of fish. What did we see? Well, first off, we had to kind of create a Flounder database, and I should say this part of the project, although it was my idea, was run entirely up in Canada at Mark Fast Lab, and he is really the expert in this. And so if you had specific questions about this, you really need to get Mark on the phone or by email. But first we had to create the Flounder database because this is not a species that's already been sequenced. So he did a de novo assembly using this program. And what came out of it was almost 200,000 sequences that were used for further analysis. These were all over 100 base pair long, a need of about almost 600. And then we used this program, DESeq, for differential analysis. And it maps these reads back onto different genes using this TOPAC program. What we found is 253 transcripts that were differentially expressed between the 2 sites. 180 were more expressed at Mauritius and Shinnecock, and 73 the other way around. This pie chart shows the overall distribution of the different types of genes expressed, and you probably can't read it. I can't read it without my glasses. But normalized for the fold change in the expression, the ones that really came out on top were ones involved in energy metabolism and glycolysis, as well as structural genes. And then there were a whole suite of other types of gene ontology groups that were affected at lesser levels. These are all around 8, 9, 10%, as well as some other transcripts of interest that weren't, there were a lot of different ones expressed. And of these, 57% had functional annotations from TLES genomes published. So, and 50% of those were from flatfish. So we thought this was pretty good. Examples of what we saw, there were 27 factors in glucose metabolism. Some examples here, 25 different factors associated with extracellular matrix and wound healing. And also some other important transcripts that we're particularly interested in. This glutathione S-transferase, this is a conjugating and sort of antioxidant type enzyme. And then there were a number of genes associated with immune and regulatory signaling shown here. And it also turned out there were 10 genes that were just unique to Mauritius, which was kind of interesting. From the differential sequencing project, we then wanted to choose individual genes for quantitative analysis by quantitative PCR, and we picked these based on the ones that were differentially expressed. Also, genes that have been shown by others. Pete, I believe Straub in New Jersey did a subtractive hybridizationing little project with Winter Flounder in 2004. So we looked at some of his genes. We tried to target genes identified as responding to stress and contaminants that are starting to come out of the big European flounder sequencing and expression studies, multi-million-dollar studies, and genes found to be responsive to contaminant exposure like CYP1A and Vitelogenic and other people's studies. Mark first validated the RNA-seq data by the quantitative PCR data by looking in individual fish that made up those pools to see what quantitative PCR showed. And 2 genes that were not differentially expressed were examined. I tried to color code them as to what they do. So the green ones are involved in contaminant, the red ones are involved in immune response, and the green ones are involved in energy metabolism. And then they also looked at a variety of the ones that were differentially expressed. And here are the data for that, showing the RNA-seq data in red, and then qPCR, both unnormalized in gray and normalized to a housekeeping gene, elongation factor 1 alpha here. And the bottom line is the qPCR data showed different, and these are fold changes in expression, went the same direction as the RNA-seq data. This is very good. And in most of the cases where the RNA-seq data was significantly different, the qPCR data was as well, which is also good. So I don't have many minutes left, and we don't have all the data, so I don't want to make any grand conclusions on this stuff. This represents only about half the data sitting in Mark's lab. The rest of it's been run, but we haven't seen it yet. He's still working with it. But preliminary, with big letters, there do appear to be some spatial patterns and expression between sites, and not shown here, but over time and over size. So here's some of the genes involved in energy metabolism, and you can see Shinnecock here compared to some of the other sites seems to be at lower levels. These are 2010 and 2011 data. The higher levels may indicate a drain on energy reserves and the need to mobilize more reserves. This is particularly evident in the glucokinase here. These are some immune response genes, pleurocidin and hepcidin. Again, the appearance possibly of some changes, Mauritius and Shinnecock, and maybe some others, but again, the data is incomplete, and there hasn't been any statistics done on it yet. This is cytochrome P450, not seeing much there, which actually matches perfectly with Jamie's data. But interestingly, this protective conjugating enzyme, glutathione transferase, is being differentially expressed. So that's kind of cool. So very quickly, summary of findings we have to date. Really no significant spatial differences in mortality between sites, but there does seem to be a hint of kind of a west to east pattern. Clear differences in environmental conditions in the bays. Cage mortality seems to be related to hypoxic periods. Predator mortality does not dominate, at least in 2 of the 3 sites. Preliminary analysis do not show large site-specific differences in condition, but overall condition seems to be related to season and most likely temperature and oxygen. The otolith analysis indicates prolonged spawning, possibly multiple cohorts, and strong signals in growth, but again, not large base-specific differences. Microsatellite data, on the other hand, indicates a high level of inbreeding and the separation of populations and very low genetic diversity between the sites, which is not a good thing for Flounder. And the sequencing project, still sort of in its infancy, is showing differential expression among key energy metabolism, protein dynamics, I didn't show you these, wound healing, immune response, and contaminant detoxification pathways. And there are clear site and season and age-specific differences here, but I really can't say anymore until we have all the data. So left to do, we've got to get that data from Mark, and finally let Mike loose on building, starting the multivariate analysis and trying to build a model to see how all these things relate to each other. So what's the big picture? Well, this was sort of our conceptual paradigm going into this study, that we had all kinds of observed habitat conditions, both physical, anthropogenic, and ecological. We would look at population health measures as well as population structuring. There would be the interplay with the individual indicators, both at the cellular and physiological level, and hopefully we will be able to build a model that links all these things. But actually, and this doesn't portray very well, but this is Shannon's view of this thing, which I really liked, although she had it animated and it was a positive feedback cycle, and it just kept getting more frenetic and bigger and crazier, but I'm much too timid to pull off that kind of thing. But I think it nicely shows how environmental stress likely affecting young adhere mortality, potential of toxic contaminants affecting that, inbreeding also affecting this, and these are all two-way arrows here. So we could have a kind of death spiral of all these things conspiring to keep winter flounder populations from rebounding, despite the fact that fishing pressure has been significantly relaxed. Just throw this out here. The fun thing, Mark developed this BLAST sequencer. That's the extent of my humor. Program. He has uploaded all our sequence data on a searchable database, so you can either search the whole thing or just the differentially expressed one. And this would allow other people, actually anyone, to match known genes from other species to your Flounder genes, or if you were interested in doing work yourself, identify genes associated with differential expression. So that's an ongoing thing. It went live, I think, in December. And I would just like to end by thanking the lots of groups and people that help with this. Collaborators elsewhere, Chris Chambers and Victoria Talby at NIMS, the Gateway National Research Area, National Park Service. We could not have done this without New York DEC, at least some of the studies, particularly Julia, Zach, and Jen. Sorry. The NAPEG sampling was done with help from under the auspices of the East Hampton Town Department of Natural Resources, particularly Mark, who took us out for free in his boat all the time. The town of Hempstead allowed us to use their boats to do the sampling there, particularly Jim Brown. And lots of groups working with Mark Fast up in Canada. The Hospital for Sick Children is where we did all the sequencing. And he fortunately has a Novartis chair, so we were able to convert his salary money on the project to supplies and boat time, which was nice. And a huge number of people at SoMAS. I'm sure we've left a bunch of people out who helped build cages, pilot boats, catch fish, and funds for this were provided mostly through NOAA, through Sea Grant, through the Oceans and Human Health Initiative and a grant to Chris, and most recently through the National Marine Fisheries Service Salt and Salt Kennedy Project, and as well as funding from the New York State Department of State. And at least some of this work was done using the facilities of the Marine Animal Disease Lab. Thank you. So I'd be happy to try to answer any questions. Yeah?
Yeah, Andy started off the seminar talking about feminization, and then you got into the fact that the fish were generally sick, or you think they're generally sick, but they're not sick at different parts of the island.
Well, actually,
what happened to feminization?
They're not. If I had to make a statement without all the data in, I would say that these fish, condition-wise, don't seem to be terribly sick. The population is in trouble because of the likely huge amount of inbreeding, most likely because of just extreme low population numbers.
Okay,
but getting back to feminization,
yeah, right.
In Jamaica Bay, and that early data was from a while ago now, we saw clear evidence of increased numbers of females. Now, that's not necessarily bad. You talk to Dave Conover and a lot of fisheries people. Some in aquaculture, they try to boost the number of females. I mean, they're the ones putting out the baby fish after all. But there is the possibility of problems. Altered vitellogenin levels can lead to kidney problems. They can lead to, and the ovotestes can lead to poor sperm quality. So at some level, you're going to impair reproduction with that kind of thing. What we don't know yet if it really does, and in part because there are so few fish, reproducing fish there, it's going to be really hard to tell. Trying to continue to track vitellogenin has been something I'm very, very interested in, and we are measuring vitellogenin in the big sequencing project. So we will be able to see at least if gene expression for that protein is altered between these sites, but we don't know it yet. And most likely, certainly from the data I've shown today, there aren't huge, hit you in the face patterns really between almost anything we looked other than the level of inbreeding in the small population size. But what will be really interesting to see on an individual fish basis how all these factors co-vary with each other, and you won't be able to see that when we run the PCA and do multivariate analysis. And maybe nothing will come out of that. but there is like nothing left of these fish. I think we sampled every little bit. And you know, these guys did not die in vain. This is a pretty extensive list of parameters we looked at. It's actually really interesting. The European Flounder studies papers are just starting to come out in the past year or two, and this is a huge multi-country, you know, all hands on deck kind of effort, and they are measuring a lot of the same things with a lot more money and a lot more people. And it's actually our hope, I've talked to Mark about this, is comparing some of our sequence data with theirs. It's a different flounder species, but there should be a lot of similarities. Now they have the advantage that they had a big budget and did a lot of contaminant analysis as well, and that was the one thing we couldn't do at all. Yeah, awesome.
Whether you consider sex in those immunity studies, whether on the mucus immune factor, but also on gene expression, because like in human, female are usually stronger from an immune standpoint, and that may confound. So you may have direct effect of feminization or of contaminant on reducing immunity, but indirect enhancing immunity through feminization.
Yeah, well, and as you know, these immune parameters are very fluid, and they can go up and down in very, very short time frames. We had, you know, we have whatever time frame is when we caught them, so we can look over age and season, but we can't do. When we proposed the study, we had planned on collecting fish into the fall when they're big enough to sex. On most of these sites, there were no fish to be found from August on. Certainly in the most impacted site in Jamaica, I think mid-July on, there were no fish left to catch, and we tried hard. We fished everywhere. We went in the trenches. We dug up bodies. I mean, there were just no winter flounder left there. So we can't sex any of the fish in this study. Now, what I would love to do, and I need to hook up with somebody who's a better gene jock than I am. Winter flounder, unfortunately, is not one of those fish that either has reliable secondary. Well, not the young ones. There is no easy way to sex the young fish, although I'm sure some kind of constellation of markers could maybe do that. They really, even histologically, you can't sex them until they're reliable, until they're about this big, and then you have to do kind of serial sections through the gonad to make sure you catch it, which is a huge pain in the butt. But it would be really nice to be able to determine genetic sex in these fish, because, well, for all kinds of reasons. You know, maybe the females we're seeing were, you know, genetic males. Maybe they're genetic females. But that's a project that's on my hit list of things to do someday, because it would really inform these kinds of interpretations. Yeah?
Before coming here, I was working in Korea, and we were working mostly on Phyllolectes olivaceus, which is olive flounder. And we noticed that there is a big seasonal variation in the mucosal factors involved in antimicrobial effects. And in winter, there's a different factor that causes antimicrobial effect, and in summer, another factor.
Yeah.
But they balance each other out.
Yeah.
And I think it would be interesting if you could.
Oh, it would. I mean, you know, it's and that's one of the reasons why we did the sequencing project, to be able to look at, well, you know, not to be just develop an assay for one thing and look at it. But the Pleurocidin was one that's you know, known to be important in these fish. Whether it has anything to do with that antimicrobial activity we saw, we don't know. And actually, I didn't show you these data, but antimicrobial activity and Pleurocidin expression did not correlate. So it suggests that indeed something else is affecting that activity.
We also did a lot of proteomic studies, and they're also available already.
Great.